Hello and welcome everyone to this podcast for cell biology. In this podcast, as well as the next several that I will cover in the next 3 days in class, we're going to talk about cell-to-cell interactions. If you remember, at the beginning of the semester, and the first part of the semester, we talked about molecules as well as other structures inside the cell. Then we talked about, finally, it seemed like forever, by the time we finally started talking about actual cells. We talked about many things, but in general, we were talking about functions of the cell. Different pathways in the cell, how the cell obtained energy and used energy. And now what we want to start thinking about is how multiple cells interact. And in doing so, we'll talk about cells close to each other and cells far from each other. Before we get started, though, I think it's important to talk about this term called tissues. So if you recall, we spent quite a bit of time talking about individual cells. And now what we want to do is talk about how these cells of similar types can form what we call a tissue. Later, not necessarily in this course, but you'll talk about how this tissue of similar cell types may interact with cells of a different type that form their own distinct tissue. And all these tissues within the same structure we would call an organ. Later you'll learn how that organ works with other organs to form a system, and how together that system will form the organism, and understand how all of those work together. And that's more of a physiology question. So we're still in cell biology, but it's important for us to really understand how the cells within a tissue, within an organ, and beyond, within the whole organism, can communicate with each other. And so that's what we're going to talk about today and the next several podcasts. This global communication between cells within the whole organism. But for now, for this podcast, I want to focus in on tissues. So let's give a formal definition of what a tissue is. We can think of a tissue as the cooperative assembly of cells and extracellular matrix to form a distinct multicellular structure with a specific function. I want to talk a little bit about plant tissues and animal tissues. Plants have three basic tissues: the epidermis, and this includes the outer surface of the plant. Next includes the vascular tissue. This will transport water and nutrients throughout the plant. And the last one includes the ground tissue. Essentially, these are every tissue except the two above. So except the epidermal or the vascular tissues. They can also form what we might call the filler space of the plant and provide structure to the plant. In our discussion today, we'll focus mainly on the epidermal tissues, those that form the outer covering of the plant. Okay, now let's talk about animal tissues. Animals form one of four tissues. The first two I'm going to mention are ones that we're not going to focus on today, but they deserve a brief mention. The first are the cells that make up the nervous tissues. And these will help regulate and control body functions. Of course, these tissues will make up the organs such as the brain and the spinal column. The next one that I want to mention real briefly here includes the muscular tissues. These are important in organ and really organism movement. The next two I want to spend a little bit more time talking about today are the epithelial. Epithelial tissues contain cells that line the cavities and surfaces of organs as well as blood vessels. And the reason we're going to talk about these today is that they provide a nice example of cellular junctions, which is the second main part of today's material. Connective tissues are what we're going to spend the majority of this podcast talking about. Connective tissues support, connect, as you might imagine from the name, and separate one tissue from another tissue. So the first part of our discussion today will include the extracellular matrix, and we'll define that in a moment. But the reason connective tissue is a good tissue to talk about extracellular matrix is because connective tissue is defined by its extracellular matrix. So for instance, connective tissues make up bone, okay, so now that we've talked about the different tissues, I want to talk about extracellular material as a general concept. And then we'll move into some more specific examples. Most cells, and this includes cells from bacteria to archaea, fungi, protists, plants, and animals. For today, we're going to focus primarily on plants and animals, but it's important to remember that extracellular material is made by most cells. So what is this extracellular material? Essentially, extracellular material is secreted from cells, and when it does that, it helps define the cell's shape and function. It may also help attach that cell to other cells or to a surface. Extracellular material contain two broadly defined substances. The first are fibers. These are primarily proteins and something we will call ground substance. The fibers help protect against stretching, straining, and tension. The stretchiness of our skin is due to these fibrous proteins. The ground substance are the substances that will fill the spaces in between the proteins, and they help protect against compression or pressing forces that the organism may face or these tissues may face. So over the next several whiteboards, we're going to talk about two examples of extracellular material. The first includes the extracellular matrix in animals. The second includes cell walls, primary and secondary cell walls, found in plants. So let's go ahead and get started with the extracellular matrix of animals. So the extracellular matrix in animals. The ECM is like the extracellular material. It secretes proteins and these ground substances that we'll talk about in a moment. But in general, it's going to provide structural support for the various tissues. So remember, the ECM includes the hard material made by bones, as well as the stretchy material made in cartilage, and our blood vessels, which helps allow them to expand when appropriate and our skin, which helps explain how it can be stretchy. So remember, in extracellular material, we talked about it containing two substances. One was a fiber protein, and the other was this ground substance. The fiber protein can be one of several kinds of proteins, but one of the most abundant is collagen. Collagen is a very abundant protein. It's about 25% of all proteins in mammals. There are 25, probably more, but at least 25 different collagen proteins. And we'll talk a little bit more about collagen in just a moment. The ground substance surrounds collagen, or whatever fiber protein might be present. It resembles this gel-forming substance and is made primarily of proteoglycans. And essentially, proteoglycans are proteins containing sugars attached to them. Okay, so let's focus on the extracellular material of animals that we said we're going to call the extracellular matrix. And we'll abbreviate that as ECM. Okay, so let's talk first about how this extracellular material, the proteins, the collagen, and the proteoglycans, are released from the cell. And if you remember, everything that exits the cell has to exit through the secretory pathway. So remember, we have our friend here, the ER, where the proteins will be made with the ribosomes attached. They'll form these vesicles which leave the ER. They'll fuse with the Golgi, and in doing so they will pass this protein between the different stacks of the Golgi, and eventually it will be released in these vesicles, which I, for some reason, drew very large here, but it's not quite that large. And then it is released. The protein is now released from the cell. Now, in just a moment, I'm going to talk about how this collagen protein will interact with other collagen proteins to form these very large fibers. But before I do that, I'm going to talk about a timing problem we have here. These very large fibers that we'll talk about in a moment, we don't want them forming inside of the cell. We want them forming outside the cell where they will have their function. And so the way that this is accomplished is that the protein that's actually made inside the cell isn't specifically collagen. It's a precursor of collagen, which we call procollagen. You'll come across this term, this prefix pro, a lot in cell biology. When that is used, it's talking about the immature form of the actual protein we're interested in. So procollagen is made first. It is what's made in the ER. It's what's transferred. It's what collects all those modifications in the ER and the Golgi. And what's secreted is procollagen. And what procollagen has on it are extra amino acid sequences at the N-terminus and the C-terminus. This form, as you see it here, cannot interact with other collagen proteins. By itself, it's rather useless. But it's really important it exists in that form as it goes throughout the cell, throughout the secretory pathway, because we don't want the collagen fibers that we'll talk about in just a moment forming inside of the cell. So how do we go from collagen, or procollagen, to collagen? What happens is that there is a protease that will degrade these ends of procollagen. Proteases are enzymes that degrade proteins, and these have very specific targets. Only these ends of procollagen are degraded. The rest of the protein remains intact. And so what forms is our collagen protein that we're interested in. Now remember, we said that collagen exists in about 25 different forms. This is what helps define if we're looking at a bone tissue, cartilage, skin, the lining of vessels, blood vessels. Each of these tissues have very specific and unique collagen proteins. And we'll come back to that general idea in just a moment. But I want to finish talking about how we're going to turn this into a more complex structure. Collagens of different kinds, or it could be the same kind, it could be 3 of the exact same collagen, or they might be 3 of different kinds of collagen. But after they've been processed, what will happen is you'll have one collagen protein here, and I'm going to draw them in different colors here just to signify we're talking about 3 different collagens. But remember, they could be the same from the same gene, or they could be from 3 separate ones. But they will always form this triple helix, all with their N-terminus at this end and their C-terminus over here. So this is what we would call a triple helix. And this is the fundamental form of collagen. It can interact with itself. And then in that case, multiple forms of this triple helix together will form a collagen fibril. And these collagen fibrils can interact and form a very strong structure called the collagen fiber. Now, I didn't talk about it yet, but we could also have proteins going through the secretory pathway that form the proteoglycans. And because they're being processed through the Golgi, remember, the proteoglycans will contain essentially this core protein with several sugars attached to it to make up that mature proteoglycan. Now I want to talk real briefly about the 2 factors that are important in determining if we're looking at bone, cartilage, skin, vessels, or what kind of extracellular matrix we're dealing with. So these 2 factors include the amount of extracellular matrix, so the overall amount of collagen and proteoglycans. This can help define if we're talking about bone, cartilage, skin, or vessels. So for instance, bone and cartilage will have more ECM and less cells. In contrast, skin will have minimal, not very much at all, ECM, extracellular matrix material, but more cells. So that's the relative amount. The next factor that is important in defining which tissue we're talking about is the composition. Or we might think of this as the type of collagen, because remember, we have 25. So for instance, in tissues like lung tissue, the ECM will contain proteins that are more elastic, because we need that ability for that tissue to expand with every breath and then to relax again when we exhale. So much more elastic proteins. In fact, one of them appropriately is called elastin. And of course, bone is going to have an ECM that is more rigid. So the amount of ECM relative to the amount of cells is important, but also which kinds of ECM proteins are being made is also very important. Together they help define which tissues we're looking at. Now, even though the extracellular matrix, the material of the extracellular matrix, is secreted from the cell, it just doesn't flow around her randomly. It's still connected to the cell. So let's remember that point here. This ECM out here is connected to the cell. In that way, there's communication between the cell and the external environment, as well as communication between this cell and its neighboring cell. So how is it connected? So let's spend a little bit of time talking about that now. And to do that, I'm going to introduce a few more proteins for you. First, I'm going to show our trusty collagen here that we've already talked about. It could be in a fibril or a fiber. At this point, it doesn't matter too much. All right, so how is collagen going to interact with this cell? And it does so using these proteins, which I'm going to call integrin. Integrins are on the plasma membrane of a given cell that's going to interact with collagen. On the inside of the cell, these integrins are going to bind to actin. Now, these integrins can't bind directly to collagen, but rather they will interact with other proteins that in turn will interact with collagen. And we'll call these proteins lamin proteins. It's kind of an unfortunate name because sometimes they get confused with the lamina, which are the proteins of the intermediate filaments in the nucleus. So don't get that confused. These are separate kinds of proteins that are on the outside of the cell that help these integrins attach to collagen. Depending on which laminin you're talking about, they can also help interact with other parts of the ECM, like the proteoglycans. So we may have proteoglycans, for lack of a better way of drawing them, I'm just going to draw them like this, that are surrounding these collagen molecules or proteins. But in this way, there's a direct way that the cell is communicating with this extracellular material, and since it's linked to actin here, when the actin filaments move, they can also control the movement or the lack of movement of these extracellular matrix proteins. So these connections that I'm talking about here are important for a few reasons. It holds it in place, this extracellular material, so it doesn't just randomly float around, and it connects with adjacent cells. I didn't draw it here, but these collagen molecules could just as easily be linked to these lamins of adjacent cells, and vice versa. In that way, cells within the connective tissues can help function together as one unit, or the tissue itself. That's all I want to say about animals, extracellular matrix. Next, I want to spend a little bit of time talking about plant cell walls. Okay, let's talk about plant cell walls. These are extracellular materials produced by most plant cells, almost all plant cells. We don't call it extracellular matrix. That's a term that we use to describe this material found in animals. In talking about plant cell walls, we're going to talk about both the primary and secondary cell walls. So let's first talk about primary cell walls. The primary cell walls form as new cells are made, and they will continue to exist as that cell, that plant cell, ages and matures. These plant cell walls also must contain a fiber component and a ground substance. The fiber component is a long strand of cellulose. And we've talked about cellulose before, so we know that cellulose is a polysaccharide found in plants. So this fibrous component is not a protein like we would see with the extracellular matrix. So multiple cellulose strands bind to each other by hydrogen bonds to form microfibrils. So we may end up with some fibrils forming like this. So if you can think of this as three-dimensional, so below this here, we're going to have microfibrils going this way. These are connected, each of these cellulose, remember, are connected with each other through hydrogen bonds. But together, this is a microfibril, as is this. Now these microfibrils are connected with each other with this ground substance. And these proteins here, the ground substance, is better referred to as gelatinous polysaccharides. And one major one is called pectin. So we might want to call these pectins. In this way, layers of cellulose are interconnected with each other using pectins to form a very strong and stable structure or cell wall. Now I want to erase a little bit here because I want to talk about a special feature of these pectins. These pectins are hydrophilic. So let me write that here. I didn't leave myself much room, but hydrophilic. And because of that, these pectins interact very well with water. And because of these pectins that are separating these tough structures of the cellulose, these microfibrils, this water in between them, because again of the hydrophilic nature of pectins, always makes sure that the plant cell wall is moist. It helps hydrate this area. The next concept I want to talk about is something called Turgor pressure. It's established because of this primary cell wall. So let's begin by drawing a plant cell. And remember, we just talked about that cellulose that is outside here, and how the pectins here hold them together. And remember, those cellulose molecules make up the microfibril. And remember, we said that these pectins being hydrophilic, associate with water. So there's a lot of water on the outside of the cell. And it will continue to attract more water because of the hydrophilic nature of these pectins. So what we're saying here is there is less water inside the cell. There's more water outside the cell. And because of that, through osmosis, water will be pushed inside the cell. So let's remember that term here, osmosis. And so let's remember that osmosis is that movement of water from a higher concentration, that is from outside the cell in this case, to a concentration of water that is less, which is inside the cell in this case. Now, because of all the water coming inside the cell, this increases the cell's volume. It begins to push along the outside of the cell. The overall cell shape doesn't change too much, and the cell can't necessarily get bigger, and as a result, it pushes against the cell wall. And that is what we refer to as Turgor pressure. Now, as an adult plant, the overall size of the cell doesn't change because of Turgor pressure. It just kind of swells. So I guess it gets a little bigger, but it just kind of, the overall shape of it is about the same. But what about young plant cells that need to grow and need to get bigger? Let's talk about that for just a moment. We'll leave everything else down here the same. So I'm going to draw a smaller plant cell, a younger plant cell, one that is going to grow. But as long as it's connected to these microfibrils, the space that it's allowed to grow is limited. So what happens in this young plant, it will release a protein called expansin. Expansins, once secreted from the cell, will come along here and break the hydrogen bonds holding these microfibrils together. In doing so, it's released some of that. In doing so, it kind of expands this space a little bit, so that as water goes into this cell, the cell can now, in addition to swelling like we see before, it can expand and grow. So that young cell is now able to take in that water and grow because it's not held in place as rigidly by the microfibrils, because expansin has broken those hydrogen bonds. So the take home here is expansin is secreted, breaks those hydrogen bonds, allows that water, when it comes into the cell, to expand, allows the cell to expand and get bigger. Okay, now I want to move on to the secondary plant cell wall. Alright, let's talk about the secondary plant cell wall. The plant secondary cell wall is secreted as the plant matures. So if we have our plant here, and we'll draw the primary cell wall here, we'll just kind of draw it generically here. The secondary cell wall of that aging plant, the mature plant, will be produced in between the primary cell wall and the plasma membrane of the plant cell. Depending on the plant cell type we're talking about, this secondary cell wall can have different functions. For instance, it could be that waxy part of a leaf, or it might help form that very stiff, strong material on a stem or tree, essentially the bark. So if you think about it, this is extremely important because it provides the ability of a tree like this. Sorry for the lame drawing of a tree, but it provides the strength necessary to withstand gravity. It certainly protects it against various external pressures, but without this secondary cell wall, this tree could never grow as tall as it needs to grow. Even if we're talking about a small tree, gravity, you would predict, would just pull it down. But the strength of this secondary cell wall allows the tree to grow to great heights and withstand this pressure of gravity. So what makes it so strong? It's strong because of an interconnecting network of cellulose, lots of cellulose, plus this new protein called lignin that we haven't talked about before. But lignin and cellulose is what provides this very strong material we see in plants, and it's part of that secondary cell wall. Okay, so that ends our discussion of extracellular material in animals and plants. In the next podcast, we're going to focus on epithelial cells and the way that they're connected with each other using various junctional complexes. If you have any questions about the connective tissue and this extracellular material, please let me know. If not, I'll see you in class. Bye.
Now, because my dad's down in coding, I can watch TV. Good luck.